Okay, so now the second half of the chapter 5, we will continue to discuss the chromosome. So previously we discussed the virus genome, bacterial genome, and now we will discuss the eukaryotic genome. So for the human, as we all know, that each human cell, they have 2 copies of each chromosome, and one inherited from the mother, we call it a maternal chromosome, and the other one from father, it's a paternal chromosome, right? And then both the maternal and the paternal chromosome of a pair, we call it's a homologous chromosome, okay? And the only non-homologous chromosome pair are the sex chromosomes in males, all right? That would be Y, and which is inherited from the father, and the S chromosome that's inherited from the mother. So this is in the male, right? And so each human cell contains a total of 46 chromosomes and 22 pairs that come on to both male and female, and then only two sex chromosomes. And then for the chromosome, as you know, we have these 23 pairs plus a pair of sex chromosome. And hold on a second, you cannot hear anything. Let me check my. You cannot hear. Let me see. Can you hear? Can you hear me?
Yes.
Is my voice clear or just on and off?
It's clear.
It's clear.
It's clear. Oh, okay. All right. So for those chromosomes, okay. So for example, the smallest one is in chromosome 21. Chromosome 21 has the 46 million base pair of DNA. So that'll be 46 megabyte. And this is equivalent to the 1.4 centimeter long of the extended DNA. And in order to fit this DNA, right, the DNA like this, right, the smallest one is the chromosome 21, which is 46 mega base, right? So 46 million base long, and now be equivalent to 1.4 centimeter, right? So the 1.4 centimeter long now be impossible to fit into the cell, so that those genome, those chromosomes must be packed, need to be packaged, right? So that they can fit into the cell, right? Okay, because at the most condensed moment of mitosis, the chromosome is 2 micrometer long. From 1.4 centimeter, you have to pack into the 2 micrometer long. So that would be the packaging ratio is about 7,000. Okay, they have to package it for 7,004. Okay, and in the cell, the packaging system is regulated by the association of a DNA with protein. All right, and this process include the packing of the DNA into the nucleosome. From here, you see the DNA double helix at 2 nanometer wide. All right, and then you pack into this nucleosome. This is the fundamental repeating unit of the chromosome. Okay, so they package this into the nucleosome, this 2 nanometer, and then this is 10, 11 nanometer, and then they will pack further into this 30 nanometer, and then they form the loop, that's a 300 nanometer, and then they compress it fall into this 250 nanometer fiber, all right, and finally this is 1400 nanometer, that's 1.4 centimeter, all right, this is a chromosome. And during this process, okay, so you package the DNA, all right? So from here, each nucleotide you can see here, right? Then when they package into this 11 nanometer, each nucleosome, they are packed about 147 base pair long DNA. Okay. And then every 6 nucleosome, they pack into this 30 nanometer higher order chromatin structure, 30 nanometer chromatin structure. And then they pack further. Okay. And then to pack these DNA into the nucleosome, basically rely on these core histones. All right. So there are 4 different type of core histone. All right. So we have H2A, H2B, H3, and H4. Right? When they form the nucleosome, the dimer of the H3 and H4, which means 2 copies of H3 and 2 copies of H4, they form this tetramer, and then they will just bring 120 base pair long DNA together, and then the H2A dimer and H2B dimer, they will join to form the complete nucleosome, and then seal the 147 base pair long DNA. All right, so that's from 2 nanometer DNA to the 11 nanometer nucleosome. And then here you can see another histone here. This one is called linker histone. So linker histone, basically the function of linker histone is to completely seal this nucleosome structure here, and also they function to bring the neighboring nucleosome together to form this 30 nanometer chromatin fiber, right? So that's why the histone H1, linker histone H1, it's sit on top of the or in the bottom of this nucleosome structure. And these histone here, they are the core histones, right? Because they have we call this core particles, right? Core particles. And for the histone, and then you can see these histone tails, all right? So later we will discuss the function of histone tails. And basically the histone tail can help to stabilize the interaction between the nucleosome and the DNA. As you may recall, the DNA is the negative charge, all right, negative charge. And so that these histone tail, they have many positive charged amino acid. So that's why they can form a stable interaction, okay? And here is just to show you the nucleosome is about 147 base pair long, right? That's core DNA. The DNA who wrap these core particle, we call it core DNA. And in between the nucleosome, the DNA here is called linker DNA, right? And nucleosome and linker DNA. And this is just to show the histone tail modification, but I will just save this until chapter 3, all right, to discuss this histone modification, okay? And here just to show you 5 different classes of the histone, right? So we have this linker histone H1. The molecular mass is about 21 kilodalton, and 29% lysine, 20% 1.5 arginine, so it's about 30%. The lysine arginine is important is because they are positive charge. So they are responsible for interacting with the interaction with the DNA, right? Lysine and arginine, because they are positive, so they interact with negative charge of the DNA. Okay. And then of course there are several different histone variants, right? So basically, the histone variant depend on the stage and also the organelle, and different type of the core histone will be incorporated into the nucleosome, right? So for example, the histone H2A, that's the regular histone H2A, right? So we have 15 copies, all right? And then that's a core histones. Then we have this variant H2AX. You have 1 copy that's responsible for the DNA repair, and H2AZ, that's 2 copies. That'll be gene activation and the silencing. So this is just to show you that the variant has different function. And then, so in the living cell, the nucleosome, they can be packed on top of one another. So they can form this 30 nanometer fiber, right? So they form this 30 nanometer fiber, okay? And then the formation of 30 nanometer fiber that come from the histone tail interaction between the neighboring nucleosome and also the presence of linker histone. And there are two different model for the 30 nanometer chromatin fiber. One is called a solenoid model, and the other one is called two-star helix. In the solenoid model, the consecutive nucleosomes are located adjacent to one another in the fiber and folded into a simple one-star helix. And then the other one is basically just a zigzag. They arrange as a zigzag. Okay. So that's a one star and the zigzag. Okay. And then after the 30 nanometer fiber, then they can, of course, they can further package to pack this further. Then we need the 30 nanometer chromatin fiber attached to the scaffold in the chromosome, right? So they have the scaffold, and so that the 30 nanometer fiber can attach to the scaffold and then can pack further, right? So that's how they form the higher order chromatin structure. So basically, as you know from the 30 nanometer fiber, they already cannot determine how they form the 30 nanometer fiber. So for this, even further to 50 nanometer fiber, 700 nanometer fiber, and 1400 nanometer fiber, this process even more complex. Okay, but the one thing is the attachment point that the 30 nanometer chromatin fiber, they attach to the scaffold, and that place we call SAR, that's called scaffold attachment region. Okay, the SAR are only in the non-transcribed region of the DNA. Okay, so that's the, and then in the chromosome, so basically, the chromosomes are in an extended relatively condensed form at the interface. Most of the region of the nucleus, they show about 1,000-fold compaction of fiber, and some regions show the 10,000-fold of compaction. All right. So the relatively at the condensed 1,000-fold compaction region, we call the euchromatin. Euchromatin, you see the euchromatin, all right, and 10,000-fold compaction. That's here, heterochromatin, all right? And euchromatin, heterochromatin. So basically the euchromatin, they normally is less condensed. And then also the gene in this region, they are actively transcribed, okay? And heterochromatin, basically it's more condensed and it's not actively transcribed, okay? And this is just to show you these two copies of the human chromosome. Then we'll skip it. Then the next section. So basically we finish this eukaryotic genome, and now the specialized DNA sequence, right? Specialized DNA sequence in the eukaryotic chromosome. Okay, so basically there are three different types of specialized sequence in the eukaryotic chromosome. Number one is the original replication. So we learned this in chapter 2. So basically, the replication is the location where the DNA duplication begins. Okay, once the replication is initiated, then the DNA replication is like this bidirectional, right? And then they complete the replication. So that's the original replication, and then original replication of eukaryotic chromosome. They are recognized by a protein complex. It's called a original replication complex, ORC, right? That's essential for the initiation for the DNA replication, because the protein complex, they open the DNA stabilize the single-strand DNA, and allow the polymerase to copy the DNA. And the ORC complex, they appear to recruit other protein, including the DNA helicase, to the original replication, leading to the start of the replication. Okay, so that's original replication. And then the second type of the specialized sequence is the centromere. Centromere basically allow one copy to duplicate DNA and condense the chromosome, which is the sister chromatid, to be pulled into each daughter cell during the cell division, just like here in the metaphase with the centromere, all right, and the microtubule, all right, and then during the cell cycle, they will pull to the opposite side of the pole, right? So that's the centromere. During the mitosis, the centromere region of each sister chromatid, they migrate along the microtubule to the opposite pole, right? And the microtubule comprises cellular, the filamentous system, right? And it's reorganized at the mitosis, so they can connect the chromosome to the pole of the cell. And the kinetochore provide a microtubule attachment point on the chromosome. And centromere of all organisms have a defined region, which the nucleosome contains a histone H3 variant we call the SNPA, right? And the SNPA is the place where the kinetochore protein assemble, all right? In the centromere region, we have SNPA, the histone H3 variant, and that's where the kinetochore attached protein assemble. That's the centromere. And then another specialized sequence is the telomere that we discussed earlier in chapter 2, all right? The telomere is at the end of the chromosome, all right? So they define the end of the eukaryotic chromosome, and they are essential for the chromosome stability, okay? And then the end of a chromosome, that we have this distinct structure, we call this a telomere, right? Telomere, we have the G-reach sequence, and then we need a telomerase, right? Telomerase come to provide the some extra sequence, right? So that we can complete the replication of the lagging strand. So that's telomere. So that's three specialized DNA sequences in the genome.
Dr. Shen, I have a question.
Yeah.
Now, is this similar to the other,
yeah,
when we covered it in the prokaryotic cell?
No.
Where
telomere is only in eukaryotic cell. That's what we cover in chapter 2. Yeah, I believe.
Okay, so it was in only
same.
eukaryotic organisms, and that was to expand the lifespan of the organism, correct?
Yeah, correct.
Because then during replication, the strand would become shorter and shorter, and you said I think it was associated with a shorter lifespan.
Right. So that's why we need a telomerase to protect the end, to give several copy, right? So that they can, when they replicate, they will be able to replicate this region to protect the shortening of the telomere. Okay, and that the length of this telomere is relevant to the lifespan. Yeah. Okay. And then the complexity and the gene number. Okay. So the complexity and gene number, because we just finished the discussion of the genome. So for the genome, we know all the DNA sequences there. All right, all the sequences there. But remember, those sequence, right? Those sequence. I'll show you this slide. Those sequence. We have the non-repetitive DNA, moderate repetitive DNA, and highly repetitive DNA. And the genes only in mostly the majority, over 90%, they must be non-repetitive DNA. So these are the gene, okay? And so the number of the gene require for any type of organism increase with its complexity. Okay, and these to determine those genomic DNA sequence in like 20 years ago, it is almost impossible. But since the development of the NGS technology, nucleus, the next generation sequencing, then those genome sequencing is very simple and very quick, and so that we'll be able to determine the connection between the gene number and also the complexity. Normally, the minimum gene number required for any type of organism increase with its complexity. Okay, and so for example, the symbiotic bacteria, okay, it's called Nausea delta septophallicola. It's the smallest non-genome of any organism, only has 137 genes. Okay, and for the genome of a free-living bacteria, they have about 800 to 7,500 genes. Okay, and so basically there's some connection. So as you increase the gene number, then the complexity increase. And also from the table here, you can see for those intracellular parasitic bacteria, they have 500 gene, and then free-living bacteria 1,500, unicellular eukaryotic 5,000, multicellular eukaryotic 13,000, higher plant 25,000, mammal 25,000. Okay, and the sequence of the genome, prokaryote, they show virtually all the DNA encode RNA or polypeptide. Okay, that's only for the prokaryote, so that the genome size will be proportion. Genome size will be proportion to the gene number as well. Okay, gene number and the genome size, because almost all the DNA sequence in the genome are non-repetitive, and they all encode for the gene. All right. And then the typical gene size average just about 1,000 base pair in length. All right. And all the prokaryote cell, they have a genome size but below the 1.5 megabyte, a mega base pair. They are the symbiotic. Okay, and this genome size suggests the minimum number of functions required for a cellular organism. Okay, and like I just mentioned, the smallest, the symbiotic bacteria is Nausea delta cephalicola. It's 112 kilobase genome and 137 gene. Okay. And then the relationship between the genome size and gene number is weaker in the eukaryotic genome than in the prokaryotic genome. All right, because all the prokaryotic sequence, they encode for the RNA or protein, so that the genome size is proportional to the number of genes. Okay, but for the eukaryotic cell, okay, for the eukaryotic cell, this relationship is weak. All right, so as you can see here, all right, so they have rice, all right, have 40,000 gene, all right, but the genome size is 400 megabase. And for man, we have almost 3,000, almost 3,000 megabase, but only 2,500 genes. So the connection is weak. And the reason is because there are many repetitive sequences in the genome. That's why that make this more complex. Okay, but for the prokaryotic cell, it's straight, it's simple. The number of the gene and the size of genome, they show the positive correlation. Okay, and here just the table to show the genome size and the gene number. So this is smallest, all right, smallest the symbiotic bacteria. So it's a 0.11 megabase and 137 genes. Okay. And then the features of the genomic sequence, right? So as I mentioned that in eukaryotic cell, the genome size and the gene number, the correlation is weak. That's because in eukaryotic cell, you will see many repetitive sequence. So in the genome, we normally will see non-repetitive DNA, moderately repetitive sequence, and the highly repetitive sequence. Okay, the non-repetitive DNA consists of sequences that are unique and they are present only once or at most a few times in genome. So this DNA include sequences that encode proteins and also the lot of DNA without unknown function. And the genes that are present in a single copy constitute about 25 to 50% of the protein encoding genes. Okay, and some genes are present in several similar but not identical copies. All right, so they are clusters of related genes with similar nucleotide sequence and are collectively referred to as a gene family. And the most gene family likely originated from a single ancestral gene and that duplicate to produce two or more identical genes, right? In the next section, we will discuss the genome evolution, right? And so those are identical genes likely diverge in a nucleotide sequence through time to produce related functional gene. So the proportion of the genome occupied by the non-repetitive DNA varies widely among the taxonomy groups, and the length of the non-repetitive DNA tend to increase with overall genome size. Okay. Remember the increase in the genome size for higher eukaryote, like plant and mammal, that usually reflect the increase in the amount and proportion of repetitive DNA as well, okay? Not just the non-repetitive DNA. So that's non-repetitive DNA. And then for the repetitive DNA, so basically we have this sequence present more than one copy in each genome. So we divide these two different types. So we moderately repetitive sequence and the highly repetitive sequence. Okay, moderately repetitive sequence, basically we have a relatively short sequence that are repeat typically 10 to 1,000 times in the genome. And normally we can divide that into two types of repeats. So we have tandem repeats, which mean appear one after another and tend to be clustered at particular location on the chromosome. And the size of tandem cluster tend to be highly polymorphic, which with wide variation between the individual, all right? And this can be used because this variation can be used to characterize an individual genome in DNA fingerprinting, all right? So this is the short tandem repeat, STR, and this has been used in the forensic science, very, very common, okay? Because it depends on the repeat number. Everyone has different repeat number. So that's why it is easy to use these to identify the individual, okay? So that have been used for DNA fingerprinting. It's a short tandem repeat. And the other repeat we call interspersed repeat. Basically, the interspersed repeat, they scatter throughout the genome, okay? And although the moderately repetitive sequence are non-coding sequence, some moderately repetitive DNA does include some duplicate gene. So for example, the ribosomal RNA. And one of the non-coding interspersed the transposon. They are the short sequence, all right? They are a short sequence of DNA that have the ability to move to new location in the genome and make additional copies of themselves. And this DNA are transposon that sometimes we call it a selfish DNA or junk DNA. Is because they propagate themselves within the genome without contributing to the development and functioning of the organism. Okay, that's the non-coding interspersed transposon. And one of there are two type. One, okay, we have this short interspersed element. It's called SINE. All right, here SINE. That's short interspersed element. It's called SINE. They are less than 500 base pair long. They can be presented 500,000 times or more in the human genome. Okay. And the best characterized human SINE sign is a set of closely related sequence we call ALU family. Right, ALU family. They have basically ALU family is 200 to 300 base pair long and dispersed uniformly throughout the genome, both between and within the gene. And the importance of ALU family is their potential for transposition within the genome, which is related to the chromosome rearrangement during the evolution. And the SINE sign basically constitute about 13% of the human genome. Okay, 13% of the human genome. And the other group we call it a long interspersed element, right? So this is short interspersed element. This is long interspersed element. Okay, they represent a shorter category of the repetitive transposable DNA sequences. Okay, these LINE9s are usually about 6 kilobase long and maybe present 850,000 times in the human genome. The transposition mechanism of the 9 is similar to the used by the retrovirus. Okay, that's the signs and 9. And this is the tandem repeat that we just discussed in moderate repetitive sequence where this short tandem repeat are used for the forensic science. And this is microsatellite with 2 to 5 base pair repeat, right? And then you have a different size, 10 to 100 units. And then for the mini satellites, that's a 10 to 100 base pair repeat, and the array of 0.5 to 30 kilobase. Okay, the important one is variable number tandem repeat VNTR. Okay, it's a 15 to 100 base pair long, within and between genes. Okay, that's the mini satellites. And then also we have a highly repetitive sequence. So the satellite DNA is the highly repetitive DNA. It consists of a short sequence repeat in a large number of times and carry a variable AT-rich repeat unit. They can form arrays up to 100 megabase. Okay, that's the satellite DNA, and also the satellite DNA located heterochromatin region and are found mostly in the centromere and subtelomere region in the chromosome. Okay, so the satellite DNA, you can find the satellite DNA in the centromere and also the telomere. Then the organelle genome. So we will skip this, all right? So we will not discuss this. So the next topic will be the genome evolution. Okay, genome evolution. Okay, for those genome to have the evolution, the first step you need to have is the introduction of a mutation. as we learned in the chapter 2. If a mutation does not change the amino acid sequence of the polypeptide product, it's called synonymous mutation, right? Synonymous mutation. If the mutation does alter the amino acid sequence of the polypeptide product, create a missense codon or nonsense codon, then it is non-synonymous mutation. Okay, the first one, synonymous mutation, would be the silent mutation, right? And the second one, the non-synonymous mutation. So you have both missense mutation and nonsense mutation. And the first step to introduce the gene for the genome evolution is to introduce the mutation, all right? And then the next step, right, in the process to fix this mutation over the many generations, right? And then the evolutionary rate will be the combination of the mutation rate and the fixation rate, okay? And so the mutation toward a new gene structure, right, is the first step of the new gene evolution. And there are many different molecular processes contribute to the formation of a new gene. And so the number of genes presented in the genome varies by several thousand between animal species. Okay. And so also the change in the total gene number that represent the total net balance between the gain and the loss. All right. Then that will give you the change in total gene numbers. So what is the basis for the numerical difference among the taxa? I mean, for example, different taxa, you have different number of genes. The differences reflect addition of a gene and losses of genes, all right? So you can add more gene or lose more gene, and so that's how you see these difference, right, among the taxa. Change in total gene number represent the net balance between the gain and the loss. And therefore, the true rate of a gene gain in evolution must be greater than the suggested by the raw number alone. Okay. And then during this evolution process, one of the most important mechanisms, okay, to contribute to the new gene is the gene duplication. All right, gene duplication. Gene duplication. So in here, to show you the example, the duplicate. All right. So basically, during this process, they duplicate this red gene, and later on, this red gene being mutated, and so that become another new gene. All right. So that's the gene duplication. And also here, just several different mechanisms to show you how they form the new gene from duplications, right? So you can go from this unequal crossover, like this unequal crossover. So you have this homologous, right? And then unequal crossover. So that means you crossover between this one and this one, right? When you cross over, so you move this one to the top, right? So you cross over this one and this one. So that means you have this pink, green, then pink, green, pink, right? Pink, green, pink. And then the other side, you swap so that you have only one pink. So that's the unequal crossing over mechanism. And also you can have this rolling circle amplification mechanism, all right? So to duplicate the gene, all right? These are two mechanisms contribute, okay, to the duplication, okay, and also to form a new gene, all right, can come from this de novo origination. Basically, the protein encoding gene occur when the gene emerges from a non-functional DNA sequence. Previously was not a gene. Then they have a de novo origination. Okay, so you can have a new gene. So the new gene can come from the duplications, all right? The duplication, you can be this duplication divergence. So that means you replicate this red gene, and then this red gene acquire the new functions, become the new gene. Or unequal crossing over, or the rolling circle mechanism to get the duplication, right? And then the novel origination. So that means like to get the new gene from this DNA sequence that originally has no function. Okay, that's the novel origination. Or you can have this horizontal gene transfer. Basically, you exchange the gene between the genome from distantly related taxa, so that you can immediately add a new gene and function to the genome. Just like this is one organism and this second organism, then you acquire this gene from the second organism, then you acquire the new gene. So you have a horizontal gene transfer. Or you can have the gene recombination, right? So that you can form this chimeric gene. So like, for example, this one, there's a Junwei gene. So basically you have this piece coming from ADH, and these pieces come from the YMP. So this is called gene recombination. Okay. And then after you acquire the new gene, the next step is to have the new gene regulatory system, right? So you need to acquire the regulatory sequence so it can control the gene expression of the new gene. Okay, so we just explained that previously. Okay, so tandem repeated sequence considered to be generated de novo by combinational action of mechanism like a mutation, unequal crossing over, gene conversion, slippage replication, or rolling circle replication. Okay. Then the evolution of the prokaryotic genome. Okay, so the free-living prokaryote and the endosymbiotic prokaryote, they have evolved diverse strategy for living with reduced gene set. Okay, so they have the gene reduction. Okay, the gene reduction. The gene reduction basically for the free-living prokaryotic cell, natural selection would be more important because they play the role directly favor the genome reduction, all right, for free living. For the endosymbiotic prokaryote, they rely on the neutral process. So what is the neutral process? Anyone know what is the neutral process? Marina.
Is it because they don't have negatively charged DNA genes?
Can you repeat again?
I was saying because prokaryotic genes are not negatively charged like our DNA.
No, all the DNA are negatively charged.
Oh, okay, sorry.
And so the neutral
Professor, what do you
process,
mean by a neutral process?
that's why I'm asking you. So that's why I call your name. Do you know what's the neutral process?
I'm unsure what it is.
Anyone want to answer? Get extra credit.
I'll give it a shot. I mean, I don't know if I'm going to be correct.
Do I get extra credit for trying?
Yeah, you can try. No penalty.
Okay.
would a neutral process just be, I don't want to say an easier mechanism, because I know it's relatively similar.
Actually, a neutral process would be a very subtle change, and then from time to time, they just accumulate little by little. And then if you want to try to think the direction that I just from the beginning of class, I say the mutation.
Wait, are you
What
talking
kind
about antigenic shift and drift?
Genetic drift? No, we are not talking about genetic drift. I'm talking about the neutral process for the genome evolution.
Is it because so the gene transfer is only vertical when it comes to prokaryotes? Is that true?
No, we are discussing in endosymbiotic prokaryote, all right? So if you have the genome evolution, and in this endosymbiotic prokaryote, they favor the neutral process for the evolution, all right? For the evolution. So as we discussed earlier, remember you have to introduce the mutation.
Wait, Professor,
So,
is
So
so
wait,
you have to think
is a
what
neutral
kind of
process something that's not gonna like affect the DNA strand? Like it's not gonna contribute any, I guess, qualities worth gaining
very a
or
little
losing?
bit close. A little bit close. Ali, you raise your hand. Ali.
is the neutral process possibly that it's not influenced by outside forces? Like, for example, like mutagens are usually like through carcinogens or incorrect
No,
DNA replication.
no. Think about the mutation of the gene itself.
If you say a neutral mutation, I'm thinking that it's just like it's neutral. It's not really going to have any effects. Like
Okay.
it's not going to be detrimental for the organism.
You
It's
just
kind of
say
like a passive.
you just mentioned a keyword. It's not detrimental to the organism itself. Who just raised the hand? Jessica.
So, so if it's like neutral, it's not like gonna like hurt it or like improve it, if that's what you mean.
Very close, very close. So it's just like Marina mentioned, it's not detrimental to the organism itself. All right, it will not cause any damage to the cell. What kind of mutation would be very subtle?
Would it be a silent mutation?
That's right, silent mutation.
Oh,
Correct. Silent mutation
So that's when you
because
chain one nucleotide,
one you change the nucleotide, but you did not change
change the
the
amino
amino
acid.
acid. Yeah. So that's for the endosymbiotic prokaryote cell. They use this neutral process to have this evolution. So from time to time, they accumulate this little by little mutation, and finally they change the whole thing. Okay. So it's called neutral process. Okay, and so there are two different different process. Okay, for this natural selection, basically they have this streamlining hypothesis. For the neutral process, that's a Muller-Ricci hypothesis. Okay, for the gene reduction. So for the streamline hypothesis, basically the smaller genome are favored directly by selection as a way to cellular economization. Okay, so this is the most common explanation for the genome reduction in the free living bacteria. Okay, so basically the streamlining hypothesis, the natural selection directly favor the genome reduction and the low GC content in the free living prokaryote. living in the no-nutrient environment. And then for the genome reduction based on the process of the Muller-Rachel, basically the population, they undergo the constant bottleneck and no recombination, and the gene reduction occurs through the accumulation of slightly deleterious mutation, which is the silent mutation, okay? And then under these conditions, the selection, because under these the condition, the natural selection, they fail to retain the gene, okay? Therefore, you have to use this silent mutation to accumulate the mutation. All right, and then those gene will become inactive. All right, and then eventually that will delete the gene, delete that special gene from the genome. Okay, so that's the Muller-Rachel and neutral process, and this is just the two the diagram to show you this streamlining hypothesis, right, for the genome reduction through the natural selection, and this is a Muller-Rachel, okay, for the neutral process, okay. So that's it for the chapter 5. Any question?